All right, so we're going to work on this problem, and I would like to read out this problem before we start working on this problem because it does require some clarification. So the problem says given 0.1 molar solution of acetic acid, whose pK is 4.75, and sodium acetate, describe how you would go about preparing 1 liter of zero point 0.1 molar acetate buffer at pH 4. Okay, that's what the problem says. But it requires a little bit of visualization, so I'm going to help do that for you. So what we're going to do is create an image that is going to help you understand the problem. So first thing it says is you have, so the keyword here is given 0.1 molar solutions. So I want you to pay attention to this. It's a plural, solutions. That means 0.1 molar is the concentration of both acetic acid and sodium acetate that are used in this problem. And how are we going to represent this? We're going to represent this using a bottle. So here we have a bottle containing 0.1 molar acetic acid, which is our weak acid. And then we're going to have another bottle labeled number 2. The first one was labeled number 1. And in the second bottle, we have 0.1 molar sodium acetate, which is our conjugate base. And we're going to mix them, and we're going to take some volume, weak acid, which is what we call as VWA, and then V conjugate base, which is the volume that we're going to take from bottle 2. We're going to put them in a third bottle, and in this bottle, when you mix the acetic acid from the bottle 1 and the sodium acetate from bottle 2, in bottle 3, you're going to produce a buffer solution. And for that buffer solution, the total concentration of your acetic acid and sodium acetate, the total concentration of that, it has to be 0.1 molar. The total volume in that bottle has to be 1 liter. And then the pH of the solution that you're producing after you mix them has to be pH of 4. And then the question is asking, describe. So here it says, describe how you would go about preparing 1 liter of 0.1 molar acetate buffer at pH 4. Okay, so this description was needed in order to help you understand what is actually going on in this problem. So we may have to do a little bit of rephrasing of the problem. So I've rephrased it for you. So when I rephrase the question, I'm rephrasing it in this way. How much volume of weak acid, VWA, and conjugate base, VCB? So this VWA is the same as this volume here, this volume. And then VCB is this volume. How much of these needs to be mixed from bottle 1 and bottle 2 in order to produce a buffer in bottle 3 whose pH is going to be 4, and then the total volume is going to be 1 liter, and then the total concentration of both, when you combine the concentrations, the concentration has to be 0.1 molar. Okay, so that's the question. So what is given in the problem? The information that's given in the problem is, it says the acetic acid concentration in bottle 1 is 0.1 molar. The sodium acetate concentration is 0.1 in the second bottle. But you have to remember that these are the concentrations of acetic acid and sodium acetate in bottle 1 and bottle 2. So in bottle 1 and bottle 2, we don't have any buffer present. We just have the weak acid solution separately and the conjugate base solution separately. So we can say that the concentration of acetic acid, 0.1 molar, is the concentration of weak acid, and we can say that the concentration of sodium acetate in bottle 2 is 0.1 molar, is the concentration of conjugate base. We can, because that concentration is not very useful. What we care about when you make the buffer is what is the concentration of your acetic acid and sodium acetate in the bottle number 3. Okay, so the point I'm trying to make here is that, so this acetic acid concentration and the sodium acetate concentration are not the concentration of weak acid and the concentration of conjugate base that you will have in this buffer. They will be different. We don't know what those things are. We will eventually figure it out. Okay, so I don't want you to confuse this concentration 0.1 molar and this concentration 0.1 molar. We're not putting both of them in the same concentration in here. We're going to mix them in some proportion. So 0.1 molar is not the concentration of weak acid and conjugate base in bottle 3. Okay, so that's the key distinction here. We're going to use acetic acid to make the buffer. So the pKa for that acetic acid is given. And then it's also saying what the final pH should be for the buffer in bottle number 3. In bottle number 3, our pH has to be, our pH has to be 4. So the pH has to be 4, which is given over here. Okay. So that's given. So what are we trying to figure out? The goal in this problem is we need to figure out how much is the volume. How much are we taking from bottle 1? How much are we taking from bottle 2 to make the final buffer solution? That's what we're trying to figure out. Okay. So what is the strategy to work through this problem? So because it's a buffer problem, we're going to follow the standard protocol. That is, we're going to use the Henderson-Hasselbalch equation and then somehow dig deeper into this Henderson-Hasselbalch equation and then figure out what the volumes are for VA and VB. Okay, so it's going to involve a whole series of steps, a lot of simple algebra. So just bear with me, but in the end, it will become clear to you. All right. So this is the buffer equation, and we're going to set up the calculation based on the information that's given in the problem. So this is the Henderson-Hasselbalch equation. We know the pH of the solution, we know the pKa of the weak acid, so we can plug them in the place of the pH and pKa, and then solve for that constant, this fraction. The fraction that we are going to get from here is what is the ratio of the concentration of conjugate base to weak acid. First, we're going to figure that out. So I put the 4 there, 4.75 for the pKa, and then I'm going to do the calculation as we've done in our other problems. And eventually, if you remember, if we need to get rid of the log term, we have to raise both sides to the power of 10. So when you do that, you're going to end up with 10 to the power of negative 0.5 on the left side on this. And we're going to also raise this side by 10 to the power. So it's 10 to the power of log conjugate base concentration or weak acid concentration. If you watch the other videos where we did similar problems, when you raise a log term to the power of 10, they cancel each other out because they're basically opposite operations. You just end up with this particular fraction. This is going to be the result of that. So when you simplify this, you end up with the concentration of conjugate base or concentration of weak acid is 0.178. Okay, so this is an important result because from here we're going to dig further deeper to figure out what volumes we need to take from the bottle. Okay, so that's the next step. So we start with that, and then at this point we're going to really ask ourselves a question: what does this term really mean? This term is saying this is the concentration of conjugate base and this is the concentration of conjugate weak acid. What does this really mean in the context of the buffer? Whenever we use the word concentration, you know that concentration refers to molarity. Okay, so we are referring to molarity of the conjugate base and also the molarity of the weak acid. So these two concentration terms refers to molarity of conjugate base and molarity of weak acid. So because it's a molarity, we can express first I'm dealing with only the conjugate base we're only dealing with this, we're not dealing with the weak acid. So first I'm dealing with this conjugate base concentration, and because we are describing it in terms of molarity, we know that that's moles of conjugate base in the buffer divided by the total volume of the buffer that we have in the bottle number 3. So when we say total volume in liters of the buffer solution, we are referring to bottle number 3. Okay, so we're trying to figure out what's the moles of conjugate base in this buffer divided by the total volume that exists in this particular bottle, in bottle number 3. So when we say moles of conjugate base in the buffer, what are we talking about? You do know that moles means the collection of particles. So we are actually counting the number of particles of conjugate base, which is this guy right here, this bottle number 2. How many particles of conjugate base ended up in bottle number 3 is basically the moles of conjugate base. So what controls the moles of conjugate base that ends up in bottle number 3? It's the volume that we take out from here. The volume that we take out from this particular bottle decides how many particles of conjugate base are going to end up in this buffer bottle. So if you want to figure out the moles, the best way to do it is you take the molarity of whatever conjugate base that is in the bottle number 2, and we know that molarity is moles over liters, and then we're going to multiply it by the volume that we take out from here. That volume is going to be in units of liters, and you're going to multiply them together. When you multiply them, the liters and the liters cancel off, and then you're going to end up with your answer in moles. So what I'm trying to say is that if you want to figure out the moles of the conjugate base in the buffer, you have to multiply this concentration over here, 0.1 molar, by the volume that you're taking out from that buffer in liters. So that's going to give you the moles. And then that many moles are going to end up in this bottle. But in this bottle, you're going to add your conjugate base. You're also going to add your weak acid. So when you add up the two volumes, whatever that total number is, that's the number that's going to go in the denominator. Okay, so that's what you're going to put in here. So just to simply state it, we have expressed this conjugate base concentration in terms of this particular fraction right here. Okay, and we're going to do the same sort of exercise for the weak acid. If you think about the weak acid in the buffer, the concentration of the weak acid in the buffer is the molarity of weak acid in the buffer. And we know that molarity is given by moles of weak acid divided by the liters of solution. And how do we get the moles of weak acid? We know that the weak acid came from bottle number 1. So we have to take this concentration 0.1 molar, which is the concentration of acetic acid, and we have to multiply it by this volume VWA in liters. When you multiply them together, then you will get the moles, and then the moles will be divided by the total volume of the buffer solution. Okay, so now what we have done is that we started with this equation right here. We are expressing this concentration term for conjugate base and this concentration term for weak acid in terms of the volume of the weak conjugate base and in terms of the volume of the weak acid, because eventually what we're trying to solve for are these volumes, so we need to express them in terms of the volume. So that's what we're doing right here. Okay, so now the total volume, again, if you remember, when you add up the VWA and after you mix them in the bottle number 3, the total volume has to be 1 liter. So that's what this is saying. This is saying that the total volume is going to be 1 liter. So that means the denominator in these fractions, this is also going to total up to 1.0 liters. This fraction is also going to total up to 1.0 liters. Okay, so that's the equation right there. So now we are going to go further, and then we're going to just simplify this one step further. So just to recap, so we started with this equation in the previous slide, and then we eventually expressed the concentration of conjugate base in terms of this fraction, and then we expressed the concentration of weak acid in terms of this particular fraction right here. Now we're going to substitute both of these things, both of these fractions, into equation number 1. So we're going to put this in the place of the conjugate base, and then we're going to put this fraction in place of the weak acid, and then we're going to set up the equation, set up the fraction again. Okay, so if you look, so we're doing one more, and if you remember, we said that the volume term in the bottom is 1 liter, so you can see that 0.1 molar times the volume of conjugate base, which we have here, and then we have replaced this particular thing by 1 liter. So that's what we have done. And then we are also doing the same thing for the weak acid. It's 0.1 molar, which is the concentration of the weak acid, which is over here, 0.1 molar, and we are multiplying it by the volume of the weak acid divided by 1 liter. Okay, so we are doing this, we're putting the 1 liter there so that it's going to help us make the equation a little bit smaller and it's easy to write. So that's why I plugged in the volume there. So what we are going to do is take equation 2 and then plug it into equation 1, and then take equation 3 and plug that also into equation 1, and we're going to rewrite our equation in the next slide. Okay, so if you look here, we had this equation to begin with. We simplified it, the conjugate buffer concentration as shown in equation 2. We simplified the weak acid concentration as shown in equation 3. Now we're going to substitute equation 2 and equation 3 in equation 1 in the place of conjugate base concentration and in the place of weak acid concentration. So when you substitute it, what do we get? So on the numerator, you're going to get this particular term, which came from the conjugate base over here from equation 2, that goes in the top. And then this part of the fraction came from equation 3, so that is gone in the bottom. So you can see the color coordinated, the green colored thing here now is on the numerator. The purple colored thing, which we saw in equation 3, is in the bottom, in the denominator of this fraction. So we just simplify it. We know that when you have these two fractions with the same denominator, we can cross the 1 liter and 1 liter out. When you cross it out, you just end up with 0.1 molar times VCB divided by 0.1 molar times VWA. And again, we can simplify this further. We can cross out the 0.1 molar out, and then you will just end up with VCB and VWA, VCB divided by VWA. So what we are saying is we have literally simplified this expression that is shown here in terms of volume. So we're saying 0.18178 is basically equal to this ratio of volumes. Instead of expressing it as a ratio of concentrations, now we are expressing it in terms of volume. Okay, so we have this equation. which expresses everything in terms of volume of conjugate base and volume of weak acid. And if you remember, these volumes are the things that we're actually trying to figure out from this sort of analysis. We want to know what's the numerical value, what's the number value for VCB, what is the number value for weak acid. We're trying to figure that out in the due course of this calculation. Okay, so what do we do here? So again, just rewriting it so that we have continuity from what we showed in the previous slides. So our final equation that we had is this. Basically, we have 0.178 is equal to VCB over weak acid, and this is the equation that we have. Now we are going to do further simplification of this equation. What we do know is that the relationship between VCB and VCA. We do know that when you take VCB and VWA together, VWA plus VCB totals up to 1 liter. So we can do a simple arithmetic here. We can just rearrange this equation so that I can say VWA is equal to 1 liter minus VCB. So this is just simple rearrangement of equation. Why am I doing this? The reason I'm doing this is because I want to express VWA in terms of VCB so that everywhere I see VWA in this equation, I can actually substitute this equation 5 into equation 4. So I'm going to take whatever I have in equation 5 and substitute that into equation 4. So I'm going to do that. When I substitute it, you're going to end up with this type of equation. Now, why should we go through it this way? The reason why we need to go through this way is because if you look at this equation, there are two unknowns. This is an unknown, this is an unknown. We don't know what their values are unless if you have only one equation and if you want to solve for the unknowns, it's impossible to solve it if you have two unknowns in the same equation. So it is better that you have two sets of equations in order to solve for these two variables. The second equation that we came up with that we used to do this simplification is this equation 5. We used equation 5 to express our VWA in terms of the concentration of conjugate base, and then we plugged it into this equation. Now it's just a matter of doing simple algebra and then solving this particular problem. So how are we going to solve this? So we have this equation here. So one thing we can do is we can multiply this side of the equation by 1 minus VCB. We can multiply this by 1 minus VCB, and then we can also multiply this side of the equation by 1 minus VCB. So if you multiply this, we know that we can cancel out this 1 minus VCB with this 1 minus VCB. That's going to cancel out. So you will end up with this particular equation right here. So actually, yeah, you're going to end up with this particular equation right here. So all I've done is I've multiplied 1 minus VCB by 0.178. So 0.178 times 1 is going to give you 0.178. 0.178 times VCB is going to give us 0.178 times VCB, and that is equal to VCB on the right side. Now we have to simplify this equation. So what I'm going to do is I'm going to add and subtract this particular term to both sides. I'm going to add plus 0.78 VCB on one side and then also add plus 0.178 VCB on the other side. When I add it to both sides, you can easily simplify this equation, and it's going to look like this. It's going to look like 0.178 on the left hand side is equal to VCB, which we had here, plus this 0.178 VCB that we added to both sides, and that's going to show up on this side. Okay, it's going to show up on this side. So this equation sort of looks complicated, but I want you to think about it like this. Let's assume that instead of thinking of this as VCB, if you think of it as X, let's say that this is X and this is also X. And if you remember, anytime you have X, we are assuming that it's multiplied by 1x. So this is almost like adding 1x plus 0.178x. When you add them together, you're going to end up with 0.178 on the left side is equal to 1.178x. It's like adding two sets of variables. And then now we're going to solve for VCB. So when you solve for VCB, VCB is going to be just 0.178 divided by 1.178. And if you take the ratio of that, because the volume is in liters, so we know that this is going to be 0.157 liters. If you convert that into milliliters, that's going to be 151 milliliters. So if VCB is 151 milliliters, what is VWA? We know that VWA is 1 minus VCB. So in the place of VCB, we're going to substitute the 0.157 from that equation. And then when we solve this, this tells us that the concentration of the weak acid is the volume of the weak acid is going to be 849 milliliters. Okay, so So at the end of it, what did we figure out? What we figured out is that this VCB value, the volume that we're taking out from bottle 2, that one is going to be 151 milliliters. And then the volume that we're taking from this bottle 1 is going to be 849 milliliters. And you're going to mix them both in bottle 3. And when you add up 849 plus 151, you're going to end up with a total of 1000 milliliters, which is the same as 1.0 liters. So we have successfully made a buffer whose pH is 4, but in order to make that buffer, we are mixing 151 milliliters of the conjugate base with 849 milliliters of weak acid in order to produce this 1 liter buffer solution whose pH is 4.